Hello friends, I am Dr. Prashant Sharma and you are watching Medicos Hub. In this video we are going to discuss about the MHC, that is also known as human leukocyte antigen. So MHC is HLA. MHC means major histocompatibility complex, which is also termed as HLA. So MHC or HLA serve as unique identification marker for every individual, as the sequence of MHC genes is different for every individual. Now we'll discuss what is the location of such genes. Location is short arm of chromosome 6. This is chromosome 6 and these are the long arms, this is centromere, and these are the short arms of chromosome 6, and the array or sequence of HLA genes is located on short arm of chromosome 6, which are represented here. Now these genes are clustered in 3 regions. What are these 3 regions? This is HLA-D complex. So HLA-D is having 3 genes, P, Q, R. So this is HLA-DR, this is HLA-DQ, and this one is HLA-DP. So this cluster is class 2 or MHC2 gene complex. The function of this complex is synthesis of MHC class 2 proteins, which will present the exogenous antigens to the T helper cells or CD4 cells. Now this is another cluster, C4b, BF, C2, etc. And this is TNF complex. So these 2 sets are forming another cluster, that is class 3 or MHC3 gene complex. It will not help in presentation of any peptide or antigen, but important thing is that it results in synthesis of complement proteins like C4, C2, C3 convertase and properdin and BF. Heat shock proteins are also synthesized. TNF, alpha and beta are synthesized, and the steroid 21 hydroxylases are also synthesized by this complex. Now the next complex is HLA A, HLA C, HLA B. These are collectively termed as MHC class 1 or class 1 MHC complex. It results in synthesis of MHC class 1 molecules, which will present an endogenous antigen of viral or tumor cells to the cytotoxic T cells. So these are the MHC complex or array of genes on the short arm of chromosome 6. Now we'll compare the MHC1 antigens with MHC class 2 antigens. What will happen if this is a plasma membrane and this is the cytosolic part and it is the extracellular fluid? Then if this is a MHC class 1 or class 2 molecule and this is the peptide molecule, then these molecules of MHC 1 or 2 will present the antigen towards the extracellular fluid, and these are connected to the cell membrane. So here we are presenting MHC1 molecule. This is whole MHC1 molecule. This is the plasma membrane of cell. This is the cytoplasm, and this whole is the extracellular fluid. Now we'll compare the MHC1 and MHC2 antigens. First of all, These are present on membrane of all nucleated cells except sperms. These are also present on platelets. Here the MHC class 2 antigens are found on antigen presenting cells or APCs, which are macrophages, B cells and dendritic cells. The peptide antigen is presented to CD8 T cells. These are cytotoxic T cells, while here the peptide antigen is presented to CD4 T cell, that is helper T cell. Nature of peptide antigen is endogenous, that is intracellular, which may be a viral or a tumor cell protein. Here it is extracellular or exogenous. The size is 8 to 10 amino acid, here 13 to 18 amino acid. This is the size of peptide antigen which is presented. Antigen presentation is done by cytosolic pathway. Here it is endocytic. First it has to be engulfed. Peptide binding site. Now this is the peptide binding site or cleft, which is made up of alpha 2 alpha 1 in case of MHC1 and beta 1 and alpha 1 in case of MHC2. So here it is alpha 1 alpha 2 groove. Alpha 1 alpha 2 groove. Here it is alpha 1 beta 1 groove. Now the CD4 or CD8 binding site. CD4 or CD8 binding site. In this case, CD8 binding site will be there. This green region, shaded region, is basically alpha 3 subunit. So here it is alpha 3 subunit. In the case of MHC class 2 molecule, it is beta 2 subunit. Now it participates in, when only CD8 T cells are stimulated, it means that cellular immunity is stimulated, but in this case cellular immunity as well as humoral immunity is stimulated. So this is the comparison between MHC1 and MHC class 2 antigens. Now what are the proper indications or usual indications of MHC complex? First one is, it is used for tissue typing before organ transplantation. Next, it can be also used for paternity determination. Another, MHC association with some diseases is also observed, like HLA B27 is linked with ankylosing spondylitis, and HLA DR4 is linked with rheumatoid arthritis. So this is all about the HLA or MHC. Hit the like button, share and subscribe our channel to get the latest updates and notifications.